Skin cancer is the uncontrolled growth of abnormal skin cells. It occurs when unpaired DNA damages skin cells. This triggers mutations or genetic defects, leading to damaged skin cells multiplying rapidly and forming malignant tumors. This is often caused by ultraviolet radiation from sunshine or even bathing beds. There are three common types of skin cancer. However, we are only focusing on melanomas. Melanomas is the most dangerous form of skin cancer. These cancerous growths develop when unpaired DNA damage in-skin cells, forming mutations that lead the skin to multiply rapidly and form malignant tumors. The mutation makes them divide uncontrollably. These tumors originate in the pigment-producing melanocytes in the basal layer of the epidermis. Melanomas often resemble moles, and some develop from moles. The majority of melanomas are black or brown, but they can also be skin-colored, pink, red, purple, blue, or white. Melanoma is caused mainly by intense occasional UV exposure, frequently leading to sunburn, especially in those who are genetically predisposed to the disease.
Immunotherapy, also called biologic therapy, is a type of cancer treatment that boosts the body's natural defenses to fight cancer. It uses substances made by the body or in a laboratory to improve or restore immune system function. Immunotherapy drugs are designed to expose cancer cells to the immune system in order to trigger an immune attack. Some mark cancer cells so the immune system can identify them as harmful, while others boost the immune system to work better against cancer.
Immunotherapy treatment is a type of cancer treatment that boosts the body's natural defenses to fight cancer. Immunotherapy is a relatively new type of treatment for skin cancer, and it works by using the immune system's proteins and T cells. It is used in the case for when cancer is spread to the lymph nodes and other organs, as once it spreads there, it makes surgery difficult. The purpose of immune therapy is to destroy the cancer or slowing its growth. The proteins work as checkpoints for identifying harmful cells, such as viruses, bacteria, and cancer. Once a foreign body has been identified, the body sends out a large amount of T cells, a type of white blood cell, to destroy it through a release of peripherin that kills the foreign antibody. Another method is oncolytic viruses. According to Healthline, oncolytic viruses are different. They're engineered in a laboratory to only infect and kill cancer cells. Oncolytic viruses also alert your immune system to target the cancer.
Immunotherapy may work by either stopping or slowing the growth of cancer cells, stop the spreading of cancer cells to other parts of the body, and helping the immune system destroy the cancer cells. Immunotherapy is given in a number of ways, including intravenously, which means that it's delivered directly into the vein, orally, where it's delivered via a pill or capsule that can be swallowed, topically, which is a cream that can be rubbed onto the skin, but this is only for early stage skin cancer, and lastly, intravesically, where it is delivered directly to the bladder. There are various types of immunotherapy, such as checkpoint inhibitors, which disrupt signals that allow cancer cells to hide from an immune attack. They are used to treat cancers such as melanoma skin cancer and lung cancer. Another type of immunotherapy is cytokinase, which is a molecular messenger that allows the cells of the immune system to communicate with one another to generate a coordinated, robust, but self-limited response to a target antigen. While many forms of communication of the immune system occur through direct cell-cell interaction, the secretion of cytokinase enables the rapid propagation of immune signals in a multifaceted and efficient manner. The last one is through vaccines, which are used both to treat and prevent cancer by targeting the immune system.
Stem cells are a special type of cell that have the potential to divide many times in order to produce more cells, which are further useful in growth and tissue replacement. The cells are not fully differentiated and therefore have the ability to differentiate to become different specialized cell types. There are three main different types of stem cells. Embryonic stem cells, which are taken from the embryo a couple of days later after fertilization, are potentially very useful due to their pluripotency. This is their ability to differentiate into any type of cell in the body. This is necessary for embryonic development. This type of stem cells has nearly unlimited growth potential, the ability to differentiate into any type of cell in the body. When then these stem cells are used, there is usually a much higher chance of rejection due to the genetic differences from the patient receiving these cells. Despite its many uses, there is one great issue that hinders us from using these stem cells, and that is that the removal of the cells of the embryo kills it, which is extremely unethical. Secondly, there are umbilical cord blood stem cells, which are taken from the umbilical cord after the birth of the baby, which would be discarded afterwards, eliminating any ethical issues. These types of stem cells can be easily stored to be used later on in the babies or the mother. When the stem cells are used, there is also no chance of it being rejected. Although these stem cells do not have the ability to differentiate into any kind of cell in the body. The stem cells are multipotent, meaning that they can differentiate into several different kinds of cells, but they naturally only develop to become blood cells. This source for stem cells also gives us a much more limited amount of them due to the fact that there are limited quantities of stem cells that can be found in one baby's umbilical cord. And because they do not have the ability to divide as much as embryonic stem cells mentioned previously. Thirdly, we have adult stem cells. These stem cells are taken from the receiver's own tissue. Obtaining adult stem cells is difficult as there are very few of them and they are usually located deep in tissues. Although this process does not kill the individual, these stem cells are once again more limited in their differentiation. They can't differentiate into as many different cell types as the embryonic stem cells, and they're also multipotent. They also do not have the ability to divide as much as embryonic stem cells, although these stem cells are fully compatible with the adult tissue. Stem cells are used to aid the recovery of the body after strong treatment such as radiotherapy or chemotherapy. During chemotherapy, the stem cells in the bone marrow and the malignant cancerous tumor cells are killed. Soon after treatment, stem cells are then injected to replace the damaged cells, which further aids tissue repair. However, stem cells transplants are not frequent amongst the treatment of malignant melanoma or any other types of skin cancer. Stem cell transplants are most often used in leukemia treatment, as this directly affects the blood cells.